Hello, and welcome to part two of our data considerations talks. This part two, will be of a more interactive session where we have some hands-on activities that we're going to try to do as best we can in this virtual setting. So I hope you enjoy what we have and that you learn a lot.
Hello, welcome back. I'll be walking us through our first exercise. So to begin,
now we're going to imagine that we are all allowed to travel and we have managed to go to our chosen field site. So now we're at our field site and we want to collect population data. We don't just want abundance data. Here we have our trap that we have set out, and I want you to record now all the different types of information that you would record if you were to be collecting mosquitoes from this site. And we'll see how many different fields we get from all of you. Okay, so I hope you've all had enough time to complete your list of data that you might collect from this site. And if I was here, I would collect something like the GPS location. This is something that I'd want to collect, something that my colleagues would like to know for the next time they put out the trap, something that other people in the future might need to know. And Sam, can you think of anything else that you would like to add?
I'd like to know what kind of trap it was and what kind of attractant was used, because just because I use one kind of trap, does that mean did I turn the light on? Did I put CO2 or dry ice? Did I put a chemical attractant in?
Another thing I'd like to know is how long the trap was set up for. Was this just left on for an hour and that might be why I didn't get any mosquitoes, or was it left out during the day when it's in an area where we'd be collecting nighttime feeders, or a trap designed for nighttime feeders? So knowing the time of day that the trap was set up for is another good thing to keep on record.
Yes, that's why it's a really good idea to keep track of the time or the date at least that the trap was set, and the time and the date the trap was then picked up or collected or checked if it was a trapping system. Now, some collections might be like human landing catches, and that's kind of an instantaneous recording. So you would either the date would just be the same, or you would just record one date, of course.
So now we want to look at our mosquitoes that we've caught, and we want to think about the data that we want to record about those. So we might want to think about our methodology. What method are we using to identify these mosquitoes? Are we using morphological examination? Are we using PCR? Or are we using both? These need to be recorded.
Obviously, once you made that determination, what species did you record? And it's a good idea not to use abbreviations, because while you might think it's a good abbreviation, someone in a different part of the world, or someone a few years later, when more species are identified in that area, that abbreviation might not be as useful anymore.
And another thing, obviously, we want to record the counts. We want to know how many of each of the individual species we've collected in this area, so we can see through time if this has changed and see if this was correlated to any control measures or anything in the area.
And what did you count? Did you only count males? Did you only count females? Did you not attempt to sex them, and then it's just a mixed population? You want to record that so you know exactly what that number means that you recorded.
If your study is looking at different life stages, you'd also want to record this. So you'd want to record the developmental stage. Did you collect larvae, pupae, adults? This needs to be recorded as well.
And maybe your study also involves parity or gravid status. If that's part of your study, then you're going to want to make a column for that too. Okay, ready? So the next thing we're going to do is another interactive activity. You'll find links under the video to 4 files entitled File A, B, C, and D. What we'd like you to do is pretend that you are trying to curate some data that's been collected by different people. And we'd like you to first take File A and File B and try to combine them into one common Excel spreadsheet. So pause the video now, try to combine those 2 files, and then we'll come back and talk about it. See you in a bit. Hi, and welcome back. Hope you've had an opportunity to take a look at the 2 files that we sent. And so now Sarah and I are going to kind of walk through the two files and tell you kind of the things that we see and we notice, and hope see how many of them that you yourself kind of noticed. So I'll start sharing my screen here. Here we have the file. And I'm gonna just use Excel. I'm going to use the data filter tool just so I can quickly see like what is in these columns. And I'll start first with kind of species. And some things I note, so here, the genus, we just have one letter abbreviation usually. And sometimes that's actually problematic. It is better to spell an entire species. Even if you think like everyone knows what a Gambi is, I mean, that's great. But if you were in some other country, some other context, it's actually not quite as obvious. Or sometimes the person who's looking at this data may just be an entomologist and maybe not a vector entomologist. So things that are really common to us might not be all that common knowledge. So here I see Anopheles gambi sensu stricto, but we don't see any kind of column indicating how they knew it was sensu stricto. So that's something to think about, is how they made that determination that it's SS. I see I have this Anopheles quad here. That's an abbreviation. They've abbreviated the full name of the species. This is Anopheles quadrimaculatus. I have a date column, but some questions I would have are, what is this date? Is this the date the trap was set? Is this the date the trap was collected? I'm not really sure. Like, is this just a one-night trap collection, one-day trap collection? We have no indication from there. We have a lat long, and we have a species count. So that's kind of my first impressions of this data set.
Okay, so now we're going to look at database B, and then I'm going to use the filter tool again, just to have a quick look at species. And here we can see Culex inurnata and Culecita inurnata. And in Sam's data set before, we saw that people were using abbreviations. And for us, this would have been problematic because we wouldn't have known which species it was. And the next thing we notice, we've got sexes, we've got male and female, which is good. We've got the latitude and longitude, which is also good. We have counts, and I'm just going to check, see if we've got zeros. So yes, we have got zeros. I think we also do have some blanks that are in the rows with the data as well. So zeros will have to be looked into, because is this that nothing was collected that day but the trap was out, or was it a malfunction of the trap? Or we don't know. And how is that different from a blank line, such as, you find one here? What's the difference between 0 and a blank line? We want to see the trap types, which is good. Again, we don't know if these light traps had any attractants with them. Was there any dry ice or anything? We don't know. And then we have the trap set date, but we don't have the trap collected date, which again is something we would also like to know. Was this, otherwise we have no idea how long this trap was left out for, which doesn't make the data as useful. Another thing we need to look out for is when people report Anopheles gambi. So we'll take a look here, see how they've recorded it. And here you can see it just says Anopheles gambi. We have no protocol for species identification here, so we can't be sure if they mean sensu lato or sensu stricto. For more clarification, it would be good if we had the methodology in a column here as well.
Thanks, Sarah. Okay, now you can pause the video if you want and try to combine C and D, which are just if you want a little more practice, or take some time and try to compare C with A or B.
Okay, so and data set C, there are a few things we can see straight away from here. We've got no GPS coordinates. We just have names like high school campus, and we'll just put the filter on just to see what other names we have. Here we go. So we've got Fred's high school campus, Main Street, and Site A, which are all very unambiguous with the without any supporting information. Often this supporting information is lost over the years if it's not kept with the collection data. So ideally we would like to have the GPS coordinates as part of this data set. Another thing we can see is in the notes column, it says also found 3 Toxorhynchites rutilus. This is another species, so ideally we would like that to be a line in here, well 3 lines if we're going to be, or 1 line, whatever, depends how you want to sort it out. We would like that to be here, so that will have to be dealt with. If we take another look at the species, we can see things here. A Atlanticus, spelling mistakes, so they would need to be sorted out. And we can see Anne Francisque, not sure what that is. And we can see here we've got Anopheles franciscoi, so that could be the same thing or could not be. There are a few examples which could be quite misleading for us. Again, we've got Anopheles gambisensis stricto, and we've got no morphological identification methods. We have a date, but we just have a single date. We don't know what this date means. Is that when the trap was put out? Is that when it was collected? We have no trap, so we don't even know how they were collected. And what else might we have? We have some blanks again, so we would like to know what the blanks mean in the specimen count. And do we have anything else? Let's check the dates in case there's anything peculiar in the dates. So we do have an odd date down here, which is 1915, which probably rings alarm bells, seeing as all the others are in chronological order. So we might want to investigate that one. Often people drag down the dates and cause issues. So that's just another thing to look out for. Also, opening files in different programs can also mess up the dates. It's always something good to keep an eye on.
So thanks for sticking with us. We've covered a lot of stuff in this session. But the good news is that we have a summary of all this for you that can all actually fit on one sheet. And it's something that we call the Minimal Information Standard for Reporting Arthropod Abundance Data, or MIRIAD. And in this handout, we highlight some of the pitfalls you can run into when you're structuring your data. And we also show a really clean example of what some highly characterized abundance data can look like. So we hope you'll take a moment and download that handout where we have a link below this video. So we hope that you found this session useful and interesting when thinking about how you might want to store and record data, even just for your own internal use, to maximize your ability to use it and make sense of it later when subsequent grad students, subsequent staff members, etc. We'd always encourage you to reach out to us. If you have any help, go to vectorbase.org and click on Contact Us, and I'd love to chat with you.
Okay. We hope that you enjoy the rest of the virtual Vector meeting, and we hope to see you all again soon at some point.